My privilege, again, Professor Karikó, my privilege that you accepted to be our guest today for young students and for young researchers from Tunisia and North Africa, and also from Africa, actually. And I have to introduce you in a way that our audience understand the event. So Professor Katalin Karikó was once a PhD student in Hungary, in a university called Szeged. Is it true? Szeged, yeah. So she was a PhD student, then she continued her research as a postdoc in the Hungarian Academy of Science. Then she moved to Temple University in Philadelphia. And she continued then still in the United States as a postdoc in the Uniformed Service University of the Health Science in Maryland. Is it true? This is what I found. So
yeah. Correct.
So then she moved as a research professor at the Perelman School of Medicine at the University of Pennsylvania. And it was there, alongside Dr. Drew Wisman, that she made the critical discoveries concerning the mRNA and the potential therapeutic of mRNA. Like 10 years ago, she moved to industry, joining BioNTech in 2013. This is what I found still, to BioNTech. And she continued, of course, being a professor at the Perelman School of Medicine in America, USA, and in Homeland, in Szeged University, as a professor in Hungary. And she got the Nobel Prize. She is a Nobel laureate in 2023 for her discovery on mRNA and the development of mRNA as a vaccine for, of course, COVID, but other things. We are very happy, and we welcome you here in Tunisia online. We welcome you, and we are very eager to hear from you, and later on to hear your answer to our fundamental question. Thank you very much, Professor, and please, the floor is yours.
Yes, thank you very much inviting me, and hello, all of these young Tunisian and not so young Tunisia researchers. Try to talk about something about my life, and so I came from a very humble beginning. My parents, we lived in this very small town here in Hungary, middle of Europe, and my parents had only...
C'est quelque chose à l'adresse. Pas vraiment, mais bon. J'étais invité à assister. Mais...
I have to... Please, people, close your...
microphone.
Yes.
Yes. Can you hear me now? Okay. So, my parents had only elementary school education, and but, encouraged me and my sister. She's 3 years older. I was just an average child, having fun with my sister. And I was good at school. My sister was very good, and I had to be good. And I studied geography. I went to competition, and eventually in elementary school, I was third best in the country. And I ended up in a high school, local high school. I mentioned here we sent a letter to famous Hungarians because we couldn't speak foreign language, just Hungarian. And we sent these famous Hungarians a letter, and they responded. And for me, it was important because János Serie, investigator of stress, he wrote a book about how to handle stress. And I would not talk here today if I would not read the book. and learn and follow his advice how to handle the stress. And I ended up in the University of Szeged. And the reason why I went there, it is southern part of Hungary, because there was this big biological research center there. But, you know, I immediately, as a student, I went there. So I want to be a researcher working at the bench. But the supervisor there sent me to field trip rather than working at the bench. And I imagine myself being a chemist, you know, and ended up collecting water fleas in the pond. So what I am saying this, that you never know, you have some imagination what you will do and then ending up doing something else, but it is fine. Actually, this was my first paper, which we published with this water fleas. So you just follow whatever is given nature to you, and, you know, and try to dig in, do your best. And when the lipid lab, you know, I went back in the fall by still student, these two scientists, Ava and Erne, were try to do liposome. We are in the 70s, and they try to deliver DNA to cell. And eventually, I participated in this project, like isolating the lipids, because we were behind the Iron Curtain, and actually, it was it was under embargo to get this phospholipid mixture, and we had to isolate from cow brain. And because my father was butcher, so, you know, I was not gooish about, you know, working with brain and extracting these phospholipids. Again, you know, you never know what kind of background you are coming, and it can be advantageous if you have other type of experience. Anyway, one day when I was an undergraduate in the lipid lab, just walk in Jenő Thomas, and he was heading the RNA lab, and and he asked me that whether I am interested, whether I am interested to work and in the RNA team and synthesize an RNA, which was just discovered. Short RNA has antiviral effect, and everybody was around me, organic chemist, and I had to make this synthesize this product and set up a screening lab for viral product. And that's me here when I work in this lab, chemistry lab. And János Ludwig is my fellow graduate student. And even today we work together, although I haven't seen him like decades, but, you know, we have patent together, we publish together, because it's important that you have good relation with your fellow students and learn and work together and trust each other and proceed. So this small molecule, actually, how it acts is that it, in response to double-stranded RNA, our body makes this small RNA molecule from ATP, and that this small molecule binds to RNase L, cuts up the viral RNA that was, and suppress viral replication. I have to say that during the COVID pandemic, we learned that those children who get to the emergency intensive care unit, they have, and very seriously ill, these two enzymes, the oligoadenylate synthase, which synthesize this product, the RNase L, which can be activated by this product. Actually, the polymorphism for these two protein, RNase L, OAS, is what the reason they ended up in the hospital. So, you know, I worked on it, this one in the 70s, but even today I follow what is happening on this field. So I was working in this beautiful building, and that was my dream. But we ran out of funding, and everything was good. You know, I get a daughter, was born in 82, but we had to move because I lost my job. And here we are in 85 with my husband and daughter in the United States. And I never wanted to go there, actually. Maybe I am the only researcher from Europe that didn't want to go there, but that was the option. And I started to work in Professor Suhadolnyik's lab. And compared to this beautiful building I used to work, you know, there were no windows here, and you know, very rundown lab, but I tried to do my best. And I worked on this 2,5-A research again, and we did a different kind of analogs and work with that. And but eventually we couldn't make it to be a good antiviral compound, although we needed one because and this major viral problem was the HIV in 1985 when I was there. So eventually we did a double-stranded RNA, which was a mismatched double-stranded RNA. It was known that double-stranded RNA can activate cells and produce interferon, which can interfere with viral replication. But it was very toxic, so we made a mismatched double-stranded RNA, synthesized this, and we published in our findings. Unfortunately, we couldn't save those patients. So I was, synthesizing short RNA, long RNA, double-stranded RNA, all of these things. And meanwhile, I didn't talk about the mRNA yet. Messenger RNA was discovered 61. And after discovery, just usually what happens, they characterize structurally how the mRNA looks from different isolates. And then 1984 was very important because that was discovered how to make in test tube an RNA. Then application started in animal studies, and with the first company formed in 2000, more clinical trial proceeded. So that was it. And eventually, 60 years after discovering messenger RNA, there was the first FDA-approved product was approved by. And so the scientists discovered in Harvard University that if they use the phage RNA polymerase from DNA template, they can transcribe messenger RNA in a tube. And they did actually interferon in this first test. It has a cap structure, poly-A tail, injected to frog oocyte. And when they incubated the frog oocyte, it secreted interferon, which was physiologically active. And with that, a lot of research started this in the 90s beginning. And I was mostly interested in doing messenger RNA, which codes for a protein, which has therapeutic value. Other people started to make vaccines, like influenza vaccine 93, 94. They already used self-amplifying RNA. And 95, when the first paper was published on cancer vaccine. And more was done in Eli Gilboa, who did a cancer vaccine on isolated dendritic cells, advanced the study all the way to human trial, phase 3 trial, but unfortunately, due to futility, it was stopped. So most of this, everything started here in the 90s, but somehow when I follow researchers, they never published again on the mRNA field. And one reason I thought that because it is small amount of protein was made short period of time. Now that today I met some of them, I learned that they couldn't get funding. That's why they stopped doing. Anyway, I ended up, as Mohammed said, in University of Pennsylvania, and Elliot Barnett was interested in urokinase receptor, and that's what I cloned, made the mRNA. And when we delivered the mRNA to the cell, interestingly, this receptor was expressed and it was functional, and it is highly glycosylated, post-translation modified, and the cell knew how to do all of these things. I ended up actually in this lab because, the Temple University, I was terminated. That you can have, don't think that America is, everything is that great. My supervisor wanted me to stay there. And when he learned that I want to leave, he tried to deport me. So I started work with Elliot. You always have to figure out what you can do. And when he left, I was also demoted from my position, and I was saved by a student who was met in Elliot's lab, but by that time he was working in neurosurgery. And here in my picture of my lab, I worked here for 17 years. That was my, my desk and my lab bench, and I did all of this thing, poured the gel, cultured the bacteria. So yeah, even at the age 58, I was still doing all of this bench work because I couldn't get support. Here is David. We try to work in different things for stroke research. Again, the funding was scarce, you know, no lab coat, no safety gloves, safety glasses, and so on. But you know, I have had fun there. And when David left, also, you know, I met by chance Drew Weissman, and he came from Fauci lab, and he wanted to develop vaccines. I was always interested in therapeutic use, but we did start to collaborate. And what happened is that when we did this first study, I generated the mRNA for Drew. And again, Drew couldn't get much money, you know, funding. And so we still, both of us work with our own hands, shoulder to shoulder. And when he tested out on human dendritic cells, the RNA, messenger RNA we discovered that it induced inflammation, and we didn't know about why it is. And of course, I was disappointed because I was just want to develop an mRNA, treat the stroke patient, and last things I wanted to have inflammatory nature. And so we asked the question: is all of the RNA is inflammatory? So here in purple, you can see the RNA, which I synthesized, was very inflammatory. A lot of TNF-alpha was made, but some other isolates, most interestingly, tRNA, was not inflammatory at all. And knowing that tRNA has a lot of nucleoside modification gave the idea that maybe if we modify the RNA, you know, in the nucleoside in it, then maybe we can make RNA non-inflammatory. Of course, you know, the question was which modification we have to introduce, because at that point more than 100 modification was known already. And so we have to decide, and we have to purchase modified nucleoside triphosphates and synthesize the RNA, which contains these different modifications. And when we tested out on human dendritic cells, what we found is that some of the RNA, here the conventional one is in blue, was inflammatory, induced TNF-alpha, but some of which get modification was not inflammatory, and those, when the uridine was modified, was not inflammatory. So who knew that uridine makes the RNA inflammatory? And of course, for us was important whether it is translatable, because for vaccine or anything, you know, that viral antigen has to be encoded, so the translation is important. And what we found is that when we have pseudouridine containing RNA, then we get so much more protein, 10 times more protein in the cell. So this was, I was very happy now that I can pursue the therapeutic use, and Drew was happy to follow and use it for vaccines. What we did is, at that point, you know, in the US, you know, we file a patent and establish a company, because the patent can, you know, you are the person who discovering something can pursue and develop to a product. So we wrote a grant. This was the only grant. This is a $1 million grant I received from the American taxpayers' money for small business. And that's what we started to do things. And we promised that we will use erythropoietin encoding RNA and demonstrate that it can treat anemia. We showed in animal study that when the RNA was modified, the pseudouridine, even 4 days later, could detect the protein, and the protein half-life only 2 hours. And we just injected 0.1 microgram RNA to the animals. So it's very quickly, 30 minutes later, we already could measure erythropoietin, the protein present. So it is very quickly translated. It increased the hematocrit, so it was functional. And when we injected weekly, then we could maintain high hematocrit in these animals. And it was non-inflammatory when it was pseudouridine containing RNA was injected. So everything looked so great. My daughter just got a gold medal in London Olympics. This was her second gold medal. Here is with my sister, and this is my husband and me. We celebrated. We were so happy. But all of a sudden, the University of Pennsylvania kicked me out, terminated in my position, and forced me to retire because I never bring in the money. So I decided that, okay, I am not done yet. So I went to Germany and started to work for BioNTech. No, we didn't have this fancy building. We were on the campus and a handful of people. And I worked together with Özlem Türeci and Ugur Zahin. And we did a further optimization of the RNA. And now that not a small amount of RNA, protein was made from the RNA, but a lot of protein, extended time, so it was ready for prime time. I would say here that why it is important, the mRNA is important because there are many, more than 100 therapeutic protein is approved by FDA, and this is the fastest growing therapeutics. Many things but it is very expensive. And when we would take instead of recombinant protein, we could use the mRNA, delivering the blueprint for this protein, and then the body could make it, will be speed up the process, it would make it cheaper, more affordable, everybody can do. And then of course here we ended up, you very well known that how the vaccine was developed, when in January 2020 we learned about the genomic of this coronavirus, and 150 different laboratories started to proceed and try to develop a vaccine. And Moderna already started in March the phase 1 trial, and eventually, we get the vaccine ready within a year. And of course, a lot of people thought, oh, it was so quickly this made, and first time. They didn't know about that already in 2013, 14, and 18. CureVac, a German company, already developed mRNA-based vaccine for rabies, or Moderna had 2015, 16. They have avian flu, Zika virus, CMV, nucleoside modified mRNA, LNP vaccines already in trial. But of course, it's not 200 million, but it was just 200 people were participating in this project. And I can say that today is more than 150 mRNA-based clinical trial is ongoing. Mostly is infectious, half of them infectious disease for different viruses, primary in flu, RSV, HIV, Zika virus, herpes simplex, EBV, CMV, Nipah, norovirus, monkeypox, but also bacteria like tuberculosis or Borrelia causing the Lyme disease. And Sanofi just started a trial for acne, and of course for parasite like malaria is also ongoing human trial, and a lot of news about the cancer vaccines, which can be individualized or FixVac. It could be injection of mRNA coding for different cytokines, can treat cancer, or mRNA can code for antibodies, and cardiovascular disease, VEGF-A mRNA, it was 2018, well before the pandemic, was already in clinical human trial. And there are more in genetic disease when some gene, some protein is missing and the mRNA can be delivered, and more advanced trial for autoimmune disease like myasthenia gravis, already phase 3 trial to use mRNA with CAR, chimeric antigen receptors. And for me, I say it's not just for human, but also for animals can benefit from mRNA. They already vaccinated elephants, horses, and dogs saving their lives. And what was left is I have to inspire the next generation. These books were written by others about my life, and I wrote my own book, and these are out in 14 different languages, and I try to inspire the next generation of scientists. And with that, I just stop here. I have more on vaccines, but thank you for attention.
Well, thank you very much for this wonderful presentation, very well explained, I do assume. I also do assume that we will have a lot of questions, but as I said earlier, in the registration form, the participant already asked the question, and we like to rearrange them in 6 main themes.
Okay.
And I will start maybe with the first one, which is a very technical one about the development and the mechanism. And some of the question, so what is the significance of integrating RNA biology with advanced technology, Katalin? Is it, relevant? And again, which may be fundamental, but what are the main advantages of mRNA vaccine?
Yeah, so it is messenger RNA is easy, very easy to make. You know, when we started to talk here, if I would start, I could make here on my bench. You know, you need a template. And now that you can, the template, the DNA, you can order, or, you know, most of the company can make their own. So, you know, when you have information, it's sufficient. You know, 20 years ago, if you need, let's say, a pandemic, you need a little package to arrive from the whatever place have the outbreak. But now the information is sufficient that everybody can start and make the DNA, can make an mRNA and proceed with it. I have to say, in the case of vaccine, like the coronavirus is an mRNA vaccine, mRNA virus. It has an mRNA, has 29 different proteins coded when you get that infection. But we try to eliminate 28 and just use the one which is critical to teach your immune system and won't make you sick otherwise. And so it is very, very affordable, very cheap, and that's what is very important.
Okay.
Because the best vaccine is the live vaccine, when the protein is, the viral protein is produced in your body, and then you don't get, you know, the full virus, which can maybe kill you.
Well, I have here a question. Very nice. So, is there a difference in the mechanism between mRNA vaccine and DNA vaccine, if they exist actually?
mRNA vaccine, DNA vaccine?
And DNA vaccine, if DNA vaccine exists, because I really don't know if they exist.
They were trying to develop. You have to understand that if you deliver a DNA to get translation and everything, it has to go inside the nucleus. It has to be transcribed. And so this is very, very challenging. It was very challenging. It's a smaller animal, it works better. And but you know, when it was in human trial, you know, they get more difficulties with it because they inject a cell, and most of our cell where they inject is not dividing. And so the RNA is every cell, almost every cell is translating, and that's what was more easier to use.
Yeah. So again, another technical question about stability of the mRNA vaccine. How stable mRNA vaccine are?
Yeah, so you know that we used in minus 80 at, you know, when it was formulated with the lipid, because we already had 5-6 years experience. We had aliquots of erythropoietin coding mRNA formulated, or luciferase mRNA, and we knew that when every year we took out, we know that that it was stable. So that's what at the beginning. Now that you could see that it can be lyophilized, and people are investing more research to make it, you know, that it is stable and higher temperature.
Yeah.
And, you know, because otherwise the RNA would degrade very quickly. So there are more effort is made to make it, you know, stable at higher temperature.
For these young students, for these young cell biologists, another technical question. So how do we ensure the correct folding and post-translational modification with this mRNA vaccine?
Because it is our cells are doing the translation, and I have to say that people said, oh yeah, you know, there are out of frame translation is happening. Guess what? The viral infected cells also has out of frame. or the modification that we have in the vaccine modification, nucleoside modification present in the viral mRNA as well. And so it is when it was infected the cell, it would do the same, translate the same way, the spike protein as it would be, from the virus. So that's our body is folding and making. So yeah.
Okay. So I will move to the second theme, which is technical, but about safety and long-term consideration. So they write here, is there any risk of mRNA integration into the genome and being passed to future generations?
Yes, for that one, you have to even not to get to this, your DNA, you have to go to the ovarium,
Yeah.
ovum, and so on. Listen, if somebody is a molecular biologist and would try to deliver DNA inside the cell, look at how many experiments you have to do. You have to understand that for those who like HIV and other viruses who can do that, the RNA is delivered, they have helping proteins to get from the cytoplasma to the nucleus. Okay, let's say somehow it will get there. Now that you need a special, the RNA, you cannot make a DNA unless you have a primer. In those viruses, there is a primer because there is a tRNA will have. So there are so many steps is needed because, to transcribe, reverse transcribe, you need a primer. What primer you will have here? Yeah. So the ability of the cell to do things, if somebody is familiar with, would know that.
Okay, good. I have here a lot of regarding safety, a lot of questions regarding the side effects. So people wrote about side effects and memory loss and human system weakness and relation between mRNA vaccine and increased heart attack, etc., etc. So I will summarize this in how to say it, in these side effects that we saw during the vaccination after the COVID pandemic.
Yes. I put here up one, for example. A lot of people said that the menstruation cycle was disturbed, okay? Abnormalities. Here, all of them, these are publications out in 23, 24, yeah?
Yeah.
Do you know that these people are all anti-vaccine people? Do you think that any of them is referenced the paper from 1913, 100 years ago? They already did with the other typhoid vaccination. They already had a paper. The title said that the vaccination effect on the menstruation cycle. So what I am telling you is that many things is already known, is universally true for every vaccine, because every vaccine has an adjuvant. And adjuvant is stimulating whatever is ongoing in your body. And then after activation, all the time you have immune suppression. And during immune suppression, it could be good if it is, some kind of ailment you have. Or it could be bad because people get shingles. But it was known also that they get shingles because they are older people immunosuppressed with any of the other vaccination. It is just, this so many people all of a sudden get the same vaccine and they said, oh, did you get shingles? Oh, yeah, but it happens with other vaccines also. It's not special with mRNA vaccine.
Typical bias, in a way. Okay, so I have here another question. So how long does the body retain trace of mRNA after the vaccination?
That is a very interesting question, because for decades I had to fight people. They said, oh, everything disappeared, not even have time to translate. And, I had to always argue that because the criticism came from that integrates and eliminates.
So, yeah.
In a 3-4 days, could be translated. And other thing, it is very important. You know, it is injected intramuscularly. Do you know that how many times I tried the muscle because for genetic reason? Because I am not, I didn't want to develop vaccine. I told you that I want to develop for therapeutic purpose. I tried the muscle to express something. When I injected to the muscle in animals, it always the macrophages and the immune cells that picks up these particles and they carry to the lymph node and so on. So it translates and a short period of time and degrades.
Okay, okay. Well, I have here a question that myself I had a doubt about.
Okay.
I will read it to you. So, could chemical modification of the mRNA induce vibrational signature in expressed protein, influencing immunological recognition? So, I'm not sure about what vibrational signature means here.
I don't know either, but yeah. But you know, the modification again, we discovered that if we modify this way the mRNA, when we discovered 2005, we didn't know that messenger RNA naturally has this modification because pseudouridine is uracil and ribose, but it linked differently.
Yeah.
And the more weight the same, so people did not know. We have Samer Abdallah, he raised his hands.
Samer, yeah, Samer from Palestine. Yes, Samer, please. I will allow you ask
Hello.
a question, but actually for the audience it's after my question. But because
Okay.
you're from Palestine, please, Samer.
Okay, thank you very much for this wonderful presentation, Dr. Karikó, and Dr. Muhammad for this great opportunity. As you have introduced in the beginning, it was not an easy journey in research. My question, to be in brief, what do you think about the cancer vaccines? How much are they effective to be given to as a preventive for their preventive value, or for people who are in maybe the first stage or something like in the beginning of their tumor progression?
So thank you for asking. Thank you for your kind note. So, you know, you cannot give somebody who never had cancer to say, oh, it is preventive. You can give somebody who already get cancer and they remove the cancer and they sequence what kind of mutation present, which cause amino acid change. And then they generated for this individual to inhibit, to return. So prevent return. So there are not, I am not aware that any clinical trial ongoing to give a vaccine to people that they will never get any kind of cancer. But these are ongoing, and I have to say, Samar, that most of them actually is going now in China. If people are asking me to see that whether for this and that, whether there is mRNA therapy vaccine is developed, I punch in and it is in Shanghai hospital, different things. So that a lot of trial is ongoing. In BioNTech did one for individualized cancer vaccine for pancreatic cancer, but those were that the patient had no metastasis. Now in phase 2 trial, now that whether the patient had metastasis or not, they are doing the vaccination protocol. And Moderna did for melanoma, again, you know, removing and vaccinating, identifying mutation. And the melanoma, the problem is that there are so many mutations and you don't know which is the driver, one which drives, you know, the cancer. In the case of pancreatic cancer, less mutation are there, but, you know, it seemed that it was very helpful. Those patients who were treated here in New York, you know, even 3 years later, their cancer did not return. So more knowledge, you know, AI and other things will help advance our knowledge, and that cancer vaccine will be used more.
Thank you. Yeah, thank you very much.
I will move to this next theme. So Samer, just close the mic. Yeah. So the next main question, Katalin, is the future application. So the future application, what will be, in your opinion still, what will be the most promising field for mRNA vaccine? So you showed in your presentation cancer, autoimmune disease, One Health, so animal, etc. But what will be the most, in your opinion still, the most promising application for next years?
I mean, the vaccines still, viral vaccine. In bacteria, you have to be careful because, the bacteria is not presenting much protein outside. It is, polysaccharide and other things. But still some intracellular bacteria like the TB is critical to have a vaccine. And so the cancer field is critical that, and you can see advancing the knowledge on understanding how the immune system works, as well as making the mRNA and the technologies more affordable and easy. Now that hospitals can make the RNA and just distribute in the pharmacy. And yeah, and autoimmune disease is progress. Those were mostly in CAR T cells. So in CAR T cells, the advancement is coming that they can deliver the mRNA. So they don't have to take out bone marrow for treat sickle cell anemia, but rather just deliver the mRNA in a formulated way that it will find those bone marrow cells and change it, because then it makes the technology affordable for those who are in Africa and getting, sickle cell anemia and others. And so this is the progress is going towards that. The finding a formulation that can target specific cell types and then specifically can deliver. There is difficulties to get to the brain and specific cells in the brain. And when they try to use viruses for that, of course, we can see that just recently that the patient died. So the mRNA, it doesn't have brain, would know that where to go, and then you cannot put you can incorporate some kind of ligand to the formulation, so it will bind to certain receptors and can get inside the cell. But it won't go through from one cell, get to the other cell, and so on.
Okay, I have here another enthusiast technology asker. So he said, or she said, what is the potential of combination between CRISPR technology and mRNA vaccine?
So right now, the messenger RNA coding for Cas9 is used, to correct. You heard here at Children's Hospital of Philadelphia, this little baby who born and he had mutation and ammonia accumulated in his body, and they sequenced. Immediately they made mRNA coding for Cas9, and they with this CRISPR technology they corrected, and now the baby is at home and no ammonia. So it is used, and also Intellia, the company, is using messenger RNA to code for the Cas9, the enzyme. Yeah, and this was for transthyretin amyloidosis, which is also in advanced human trial, delivering and knocking out that gene which codes for a protein that is misfolded and toxic. So this amyloidosis causing thing. And so they are using for this technology, they use the mRNA, but not mRNA as a vaccine. They used mRNA as a vaccine together like CAR T cells to make sure that the CAR T cells is staying high up. And they delivered mRNA vaccine, and for the same thing, they use CAR T, but not with vaccine, mRNA vaccine with other.
Okay, I will move then, because I will try to give some time to the audience, but just a few questions about the challenges and the perseverance. So you also showed that in the presentation, etc. But I have some questions. I will try to mismatch them or match them in one. Do you think that budget limitation, a poor country or a country in development like us that face budget, but also personal challenges, skepticism about the ideas, etc. So how, as a scientist or as a student or as a young researcher, how can we, regarding your experience, how do we face this? The lack of everything, the lack of confidence, the lack of budget, the lack of, you know.
Yeah. So in my case, actually, you know, they name after me a problem, Karikó problem, that somebody has idea, good idea, but never get money. And so what is, they realize that where is the money, the prestige and tarararam, everything, then those are, you know, getting the grant and so on. And they said that I am the periphery, where is no money, no prestige, no respect, nothing. But the freedom to do whatever I want, only if I have connection with somebody who is in the middle, who has the money. So if you cannot convince the authority to give you the money, that at least if you can convince a colleague, in my case, it was Elliot or David or who helped me to get that little money, you know, I did everything. And my husband, who is, you know, he made combs thrown away, you know, from the protein gel, who people purchased the protein gels and the polyacrylamide gel, and I collected the combs from the trash, and my husband made combs for me, and he did a lot of things, you know, help fix up centrifuges and so on. So what I can do? So if one thing the young one could go, just go away, go to another place, maybe in Europe or somewhere else, in America or wherever they can just, so they can experience then other culture, talking to other scientists. And then I mentioned that they learn more about themselves, that they can do. Oh my God, I can do that. You know, and that give them confidence, you know, and knowledge and collaborators and so on. And then when they go home, you know, they try to locally to, I don't know the resources. I know that it is difficult, but you know, they still can attract fund and money, and if they have ideas, they should proceed and try to make a company, try to make a patent, you know, keep trying, and you have to believe that you can do, because if you don't, then you say, oh, why the other people advancing and not me? So don't feel sorry. That was also Serie teaching. Don't feel sorry for yourself. Don't try to, you know, ask other people to do certain things, so that you always can figure out. You know, the internet is there. You can listen to all of the lecture, which is MIT and Harvard, and everything is free. You don't get, you know, the diploma from them, but the knowledge is there. And then you can learn. Can you imagine? I came here, you know, I mean, there were no, for me, the major advantage coming to America was to listen lectures. I mean, 1985, 2005 was YouTube was introduced. You know, up until that, you had to go to a lecture, but you can listen to anybody, and then you can read literature on the online. So there are so many things are there that you can advance your knowledge and your understanding, and reach out. You know, we reached out to two Hungarians because we couldn't speak English and whatnot. And for me, it was a time, it changed my life, because I learned that, oh, I should not, you know, when somebody, in my book, I describe my teacher, one of them hated me in high school, and he said that he will make sure that I will never get in the university. He knows somebody who will take care of this, that I won't get there. So it is, you have to realize it is your decision, my decision, to say, okay, I don't even go to the entry exam because he will take care of, or I say, I will be the best and they have to accept me. And then I learn so much more than I do without it. So that's why I say thank you when I, you know, I get an award. I thank to the people who try to make my life miserable. They made me work harder and improve me and make me resilient and don't get upset, And then when I went back in years later to reunion, I didn't went to my teacher to say, wow, la la, you see, I went to the university, I finished it. No, it is over. Just move on. People ask me also what I am telling here, the chairman who kicked me out. Throw my stuff on the hallway. This is, I don't blame a neurosurgeon that not recognizing that, oh, my idea was so great. No, he could see that I don't get any grant and I have to leave. Yeah. So don't carry any grudge against anybody. No.
Okay. Wonderful. Thank you very much. I will now give the floor to students, maybe to ask a question. So please raise your hand. So I will start with Salhi Said. Said, you can talk now. Said. Okay. So he is not. So anyone, because I have a lot of questions here. So please don't be shy and where. Kauthar, okay, Kauthar, vai. Active. You can speak, Kauthar.
Yeah. Hi, Dr. Benzema. Thank you very much, Doctor, for your excellent presentation. So I have a lot of questions, but I've chosen the important one. So I want to ask you about the using of RNA messager therapy for cancer. We know that in some cases, the cancer can be back after 5 or 6 years, and I don't know if this therapy can maintain a long term. That we know that this vaccine can activate the immune system. So can maintain a long term memory about the cancer cells when back.
Yeah, but I don't know the long term, if it is 5 or long years, we have to wait for to see that. But the cancer cell can accumulate a new mutation, and then against it was, made the vaccine was not developed against because it is a newly gained new now genes. So so I don't know. I don't know. We have to see that how long lasting the effect.
I have here a question from Professor Belkis Bouhawala, which is one of the responsible of the, if you hear us, Professor Belkis, so one of the responsible of the mRNA vaccine unit here in Tunisia, in Institut Pasteur Institute of Tunis, and she's asking about what may happen if the mRNA is irradiated using ionizing radiation. I mean, when we try to vaccinate someone and he is like doing radiotherapy or something, this could change maybe the I don't know. So that was the question. Is there any resistance?
I think it would degrade, but radiation, it depends on the depth or other thing. I don't know.
Okay.
I don't know. This is too specific, and yeah.
Yes,
okay.
yes. I have here a question from Aisha, which is a young physicist, doctor in neurodegenerative disease, and she's asking about I wrote here the ethical consideration. I mean, when we face maybe people and we have to administrate, like new vaccine or new vaccine, when the ethical consideration came.
As I mentioned, in the case of messenger RNA vaccine, already years before the pandemic happened, already they gained experience from small-scale human trials. And, when I came, I returned from Germany in 2020, March, for my husband's birthday. I came and I stuck here, because no more flight to return. I watched the television set, and then when the hospital, they landed in black trash bags were the bodies, and they were throwing to the cart there, because there are so many people died, they could not transfer them anywhere. So it was the environment was so desperate. And in New York, the Central Park, everywhere were just big refrigerator cart were there, because they had to keep somewhere the bodies. So, this is depending on the situation. When you have time and do certain things, you proceed differently. When all of a sudden you have a desperate situation, then, you proceed which is protective. And I think that the mRNA is not just, I think it is already proven, it is very safe and very effective. It is always I told the live vaccine is always the best. And right now there is one is attenuated for Ebola in Africa, they are using, Merck developed that. But in some cases, it's very difficult it takes a long time to develop a live attenuated viral vaccine.
So then I will jump on that. So you said Africa, you said effective, you said Ebola, etc. So do you think that mRNA vaccine could help in the global equity and the global health equity? Yes.
Yeah, I think affordability is important, and you can make mRNA. When they said that, oh, we want to do the conventional way, like here in America right now, everything in the egg. They don't know it is not really the egg, because it is a chicken embryo inside, and that's what, the cell is. They don't understand that the virus, you just cannot culture in an incubator. It is not like the bacteria or a yeast or something, fungi. You need cell. And so it is when all of the capacity, which is right now, which is the conventional way to generate, and you try to generate here like the corona, you cannot reach even half of the population, because it is very demanding, and there is not capacity for that. But mRNA, that in Africa they set up this BioNTech sent also this unit. You can make the RNA there, whatever is locally the problem. You can make the DNA, you can make the RNA, you can formulate. And right now, those people who are close to those who are infected can be vaccinated, or delivering mRNA, which is code for the antibody. So when the doctor has to go there where is already infection ongoing, they can deliver the mRNA coding for the antibody, and then the doctors or the healthcare workers already protected.
Okay. Makram also want to ask a question from Institut Pasteur of Tunis, I think.
Yes. Hi.
Hi.
Yeah. Can you hear me?
Yes.
Yeah. First, I would like to thank really Mohammed for this great opportunity, and let us listen to this great talk, and also to get in touch directly with Dr. Karikó, this great lady. Actually, my name is Makram, and as said, I'm scientist. I'm basically immunologist, and have a background in molecular biology. And right now, I'm coordinating the steering committee to implement this WHO program for mRNA technology transfer program in Tunis. And we are building this unit for mRNA-based vaccine and therapeutics based on this nice initiative. So my question is regarding the safety of the self-amplifying RNA first, and if you can emphasize on this.
Yeah. I can tell you, right now is already approved in Japan as a booster. For me, always was there that when you have to amplify something, you have to have a lot of building blocks. And, the self-amplifying RNA worked very well in the animals who are smaller, where there is more cell division and more nucleosides to replicate. In larger animals, it was not as effective, and in a nanogram or a very small amount, you had to give almost the same amount as you had to give from the conventional RNA. And this is similarly, you know, there are circular RNA. I have to say in 1994, people said that the circular RNA is a new thing. 1994, I already had a rejected grant on circular RNA. And for self-amplifying RNA, you have to realize that you deliver an RNA and it has to be replicating, and you need a lot of nucleosides, and a lot of nucleosides only there in cells which can divide, and the immune cells, which is responsible for like dendritic cells, macrophage, they are not dividing. And so, you know, there is, I tried actually replicative RNA also with the dendritic cells, and it's not working very well because there is not enough nucleotides, because it has to, when it replicates, it needs building blocks. So,
and regarding the safety, how safe is it? Because, you know,
I think it is safe. Yeah.
Really, yeah, because it's amplifying, self-amplifying, and there is no concern regarding the safety. Yeah.
You know, listen, there is a mRNA, the vaccine where the live viruses is given and they can replicate.
Okay, yeah, yeah.
So it is safe. It is just, I think that sometimes challenging because, you know, 260 kilodalton is the replicase. So the RNA is 7,000 nucleotide longer. So it is challenging, you know, making the RNA and make sure that, and then you have to include some plus the replicase plus whatever you targeting. Yeah. So, so that technically maybe is more the challenge to make the
If I
RNA.
can, I just have another question, just a small question regarding the injection intravenously. That's mean the systemic way, the nanoparticle charged with the mRNA. Is it that safe? Is it doable or yeah?
I mean, they are not doing for vaccine. They don't do IV injection.
For therapy.
For therapy, when we inject something IV, if it is not some special formulation, it ends up mostly in Kupffer cells in the liver. Most of this, so that is why, you know, the technology which was used also here in the Children's Hospital is easy to reach the liver and different cells there. But for therapy, then you have to target something in the liver, or liver cells can produce some protein if that's the part of the therapy.
Okay, thank you very much. Thank you, Mohammed.
Merci, Makram. We have another question from a young student, Aisha Mahweshi. Yes, Aisha.
I would like to thank you so much, Dr. Mohammed, for this amazing conference. I have a question regarding mRNA vaccines. If an individual is already infected with a particular strain of a virus, such as HPV, for example, would the administration of an mRNA vaccine after the infection still confer protective efficacy, either against the same strain or against other strains not yet acquired?
Yes. So you have to understand that every vaccine has to have an adjuvant. And in that case, the mRNA vaccine is the ionizable lipid, which, you know, if you just made a particle without any RNA, and it can use with a protein to together to generate immune response, because it is an adjuvant. And what happened, the people emailing me, like they have a wart, which caused by the virus, and they said, oh, after I get the vaccine, it disappeared. What happened is his body tried to fight the virus, and that get help because the adjuvant effect. And so when you have you're vaccinated, or you have a response and you get the vaccine, for example, for COVID, then it will help the other fight of your body when it fight against another virus or something. And so, of course, it could be beneficial. You know,
even after the first infection?
The infection, if it is the same time. So some people said that, a person sent me that my father died, get the vaccine, and the next day died, and because of the vaccine. And then later said that, autopsy showed that he was infected. So when you get some vaccination during infection, and that's what happened during the pandemic, there were no time and everybody get vaccinated and they didn't look.
That's
So what
true, yeah.
happened? Then, all of a sudden, their natural immune system had to fight. They didn't know that the vaccine is just pretend to be the infection and the real virus, and then it just could not do it. So, better understanding of all of these things. I don't say that the virus system, which was put out, 24 people claim that they get pregnant from the vaccine. And 20-something people said get syphilis, because they said, I had no syphilis before. I get the vaccine. They forget that they did something else. Just the same case for the pregnancy, So, but to answering your question is like I can say also one person said that I get cancer from your vaccine. I check my breast every night and I had nothing. I get the vaccine in my arm and the same breast, I get a big tumor the next day.
It was already there.
Yes. So, but the person didn't know that the cancer cannot grow so fast that today is nothing and tomorrow only the T cell. The T cells, which we can touch out, the lymph node, they can proliferate like crazy. And that's what happened. Her immune system fighting that cancer, but get the boost because the vaccine, the adjuvant effect, and then they started to proliferate and they fight and they recognized they took out the cancer. And I told her that thanks to the vaccine was recognized. But if the people do not understand things, like people are saying that, people don't get conceived because of the vaccine. I have to say, in a Hungarian head of this center for infertility told me that so many couples here for years, they couldn't conceive. And after the second dose, they get. Of course, not from just that, they worked on it. And she tried to get the explanation. And the explanation is that after you get the vaccination and immune boost, you always get the suppression. That's when people get the shingles and other things, because the immune suppressed state. And probably, they couldn't conceive because there was some inflammation ongoing and that was suppressed. And then they conceived. So, if the people would understand better the basic biology, and this is everybody's responsible for it, who is right now here, all of us, to tell the public about, the basic science in somehow a language that they would understand. And then they don't fear, because they fear the vaccine and other things because they don't understand.
So definitely.
Thank you so much, Doctor.
So waiting for another question, I will start with another one here. So everybody now is talking about personalized medicine. Medicine, and all this genetics difference, the ethnicity, etc., etc. So is there any extra effort to do with mRNA vaccine regarding any disease or something? And regarding this difference in ethnicity, is there any effort to do, or is there any extra stuff to do regarding that?
The ethnicity now is important. You can hear that something which worked in China did not work in Europe. But that was not mRNA related finding. But this individualized vaccination, now that the RNA making is very cheap, very quickly, so somebody who had an operation and sequencing can be done in overnight, and identifying mutation that cause amino acid chain can be done very quickly, and then mRNA can be done in a day. And right now, from the operation to get back all of these individualized cancer vaccine, I think 4 to 6 weeks somebody could get. So it is get a more mainstream, that how the technology is, everything is automated, and so that, and it is very affordable. So the advantage is that, less side effects, because I think exactly where the problem is. And that's the advantage of all of this biological medicine is, less side effects and more targeting whatever is causing the problem. So it will come, and more for other fields as well.
Yeah. So the last question from the audience, and if not, because we still receive like a written question, etc., I will ask you, lovely people, to open your camera because we will take a picture. If you can open your camera, all of you, and we will try. And if our guest, Professor Katalin Karikó, can also show us the middle.
Oh.
Yes, because we will try to take some picture. If you can open the mic, please, to take this screenshot.
Actually, the other side is only just medicine. Medal has two ladies are there. One is sick, the other is helping.
Oh, okay.
Yeah, the Medicina, the goddess of healing. Yes.
So I will take this one and second one. Wonderful, wonderful. Okay. Yes. So maybe to close this wonderful, I would say, conference and talk and questioning and reflection and science, etc. So
Yes. Yes. So thank you very much for inviting. I'm so glad that so many women is around here, and I could tell them that my example shows, that you don't have to choose between having a family and having a career, because, I have my daughter, but if somebody is here from the government, maybe somebody who's no camera there, then maybe that's the government. Please, help these women to get affordable, high quality childcare. And because that's what, thanks to that, my daughter was 4 months old and she was in the daycare, and she came out okay. She's a 2 times Olympic champion, as I just showed in the lecture. So she's fine, and she has 2 grandchildren. Now I am grandmother. And so it is, my message, which I mentioned in the beginning, is that you have to have a mental and physical health. So exercise. I told Mohammed this morning, 5 o'clock, I went to swimming pool. 1 hour I was swimming, old ladies, that's what I can do now. Prior to that, when I could run, I run 6 kilometers every morning before I went to work. 6 kilometers running. I love running. And so it is important that also you don't take seriously, don't get mad at people, learn just to focus on things, and it is so much happier. I have my book here, it is the English version. I describe that, I also was very humble beginning, and I stayed the same. That's what I am most proud of, actually, that I am not different. And I could understand all of others because I was behind the curtain. And my not a privileged family that my parents is professors or somebody's, but it is just to stay the same. And right now I am focusing on other diseases to help some people who are suffering, and so you always find purpose in life, and that's what makes fun. Yeah, that you try to, all of you want to do something which is helpful and helping others, and you have search for knowledge, and the knowledge is there, you know, on the internet. You can reach all of these knowledges there and reach out for fellow scientists, you know. You don't assume that, oh, you are from Tunisia, nobody knows, you know, where it is. You know, they didn't know where Hungary is. Just, you know, be brave and reach out and try to find, you know, go to another countries. As I mentioned, you learn more about yourself.
Yeah.
Then, you know, really, you understand another culture, and you will realize that, you know, they don't look down on you. You know, it is sometimes in the people's mind is that, you know, that you are a fellow scientist and respect. And so that's what you have to think about. And yes, so I enjoyed being a scientist.
Thank you very much, Katalin Karikó, Professor Karikó. Thank you very much. You was wonderfully inspiring for all of us. I will just tell you, Yaishik, which means in Tunisian, may God give you a good health and good life. Yaishik. We use it to say thank you very much. So again, thank you very much in my name and in the name of all my students and young researcher here. Thank you very much. Have a good day and see you in another adventure, hopefully.
Yeah, thank you. I can see all you girls here, guys here.
A lot
yeah.
of girls, of course.
Yes, great.
Thank you very much. Thank you very much.
Very smart. Yeah.
Bye-bye.
Take care.
Cheers. Bye.
Bye.